Thank you very much for the introduction. And I'm going to be telling you about DNA unknotting and unlinking by XCR-CD. So, well, it's on. Maybe I put it closer to me. Is that better? Okay. So, let me make sure that this works. Okay, it won't work. I'll do it manually. Oh, that doesn't work either. I'm sorry about that. Okay. So before even starting, I wanted to thank the organizers for such a great meeting. I know I'm not the last speaker, but I wanted to say this. It's been really fantastic. As some of you know, I've been out of service for years, so this was a really great way to come back, and it's always a pleasure to be in England. And it's been really, really great to see this mixture of mathematicians and biologists and chemists and physicists all working together. The community is growing, and it's really wonderful. So thank you very much, and maybe a round of applause for the organizers. So now I'm going to start with DNA, because I could not do it otherwise, being in Cambridge. And with a quote by Watson and Crick, where they said it has, in their seminal paper, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. And then they went on in their next paper that same year to say, since the two chains in our model intertwine, it is essential for them to untwist if they are to separate, and they do indeed untwist. Although it is difficult at the moment to see how these processes occur without everything getting tangled, and everything does indeed get tangled, we do not feel that this objection will be insuperable. So just like Ian said, if things are not resolved, if dimers are not resolved, and if catenanes are not resolved, the cells just die. So, but not all cells with circular chromosomes die, fortunately, so the objection is not insuperable, and the cell has mechanisms to solving topological problems. So the greatest example of this is replication of circular DNA, and we heard quite a bit of this yesterday, so I won't go through any details, but if you have a circular DNA molecule, I'm not drawing the circle here, I'm just drawing the replication fork, you get positive supercodes ahead of the replication fork. These will turn into precatenanes behind the replication fork, and Jorge gave a really nice talk about that yesterday. And these precatenanes, some of them are resolved quickly, but others will turn into catenanes after replication. And those will need to be resolved if we want these cells to survive. So type 2 topoisomerases will resolve these replication catenanes or links. So I'll use the word catenane or link indistinguishably. Link is a math term, catenane is more used in biology. So type 2 topoisomerases do this. In the case of E. coli, it's mainly topo 4, as we heard yesterday. So in the bacteria E. coli, in the absence of topoisomerases, it was shown in the Sherrod lab that XCR-CD coupled with the powerful translocase FTSK that Ian told us about can unlink these replication links. So that's what I'm going to be focusing on for this talk. So I've had really wonderful introductions before me, so I won't spend too much time talking about the basics of site-specific recombination, but just to remind you, the recombinases act at two specific sites. These sites are short DNA molecules. Recombinases will effectively introduce one double-stranded break on each one of the sites, recombine, and paste the sites together. These could be a double-stranded break at once, or in 2 steps if the recombinases belong to the tyrosine family and go through a Holiday junction intermediate. All that is not in this very basic cartoon figure. So the idea is you start with 2 sites, cut, recombine, paste, and you get perhaps a crossing introduced in the domain. Changes in the DNA mediated by these enzymes have important phenotypic effects, as we've seen in the previous talks. So just summarizing what Ian told us, 1 in 8 generations, the E. coli chromosome or naturally occurring plasmids will turn into big chromosomal dimers because of an error of, well, because of homologous recombination reaction that did not go properly. So the replication fork will stall, homologous recombination enzymes come in to try to solve that problem, and they make a mistake. So instead of getting you your 2 newly replicated circles as 2 separated circles, they give you a big circle with 2 identical halves, a big chromosomal dimer. So this dimer has to be resolved, otherwise we get those filamentous cells that Ian showed in his movie. And the dimers are resolved by colocalizing the 2 diff sites at the division septum. So this is the division septum. These are 2 copies of FTSK. The mechanism is not very well understood yet, but the 2 diff sites will colocalize here, and then XERC and D will be recruited to that location and resolve the big dimer into 2 monomeric molecules. So very, very important role of XER in E. coli. And just turning that into just a cartoon figure, we have 1 big chromosomal dimer with 2 identical halves, and recombination resolves that into 2 smaller, here they're not smaller, but they should be smaller, monomeric circles. Okay? Okay, so the other interesting thing, and we've also seen it in the last few talks, site-specific recombinases cannot and they can link circular DNA. Lambda integrase is known to very effectively create right-handed torus links when the sites are in direct repeats. XERCD, as Sean told us, will produce a unique topology, a right-handed 4-crossing torus link with anti-parallel sites. These enzymes can also unlink. So the first experiment along these lines done in the Sherat lab by Ip et al. In 2003 showed that if XERCD FTSK were incubated with torus links created by lambda int, so here the sites are in parallel orientation, then those were effectively resolved into just 2 monomeric circles, unlinked circles. Okay, so how do these site-specific recombinases introduce topological changes on the DNA? So if you've seen me talk in the last few years, you've always seen this figure, but I really think it's a very good way to explain to those of you who have not seen me talk what the problem at hand is. So locally, we understand the reaction. Cut, recombine, paste, done. Biochemically, it's really interesting and much more sophisticated than this. But from a topological point of view, it's just cut, recombine, paste. However, if you look at a global scale, you look at a DNA molecule here with 2 sites in direct repeat, let it float around in solution, and assume that the recombinases, say lambda int, for example, bind 2 recombination sites at some fixed point in time. So let's take 2 snapshots in time of this DNA molecule floating in solution, and in this moment, for this particular protein, 3 crossings are trapped in the domain prior to recombination, whereas for this one, only 1 crossing is trapped in the domain. Then we recombine just by cutting, recombining, pasting, identically in both cases, and we get different topologies. So this geometry or topology, well, let's call it geometry since we have unknot here, outside the synaptic complex has a direct impact on the topology of the product. This is what we want to understand. So the product topology is a direct consequence of the geometrical conformation adopted by the substrate prior to recombination. This is very important. Okay, so our goal is to understand the topological mechanism of binding and strand exchange. What is topological mechanism? Well, this is to be opposed to a biochemical mechanism, which is pretty well understood at the atomistic level. So topologically, globally, what is this enzyme doing? Using geometry and topology. Okay, mathematics of site-specific combination. Isabel told us a lot about that, so that's really really great. So just to remind you, we have a double-stranded DNA molecule with a specific recombination site. We forget about the double-stranded nature of DNA. We forget about the sequence, more or less. And we model this double-stranded DNA molecule as the axis drawn, as a curve drawn by the axis of the double helix, like here. Okay? And we may remember the location of the site and the orientation of the site. Now, if this molecule is circular, then the axis will be a circle properly embedded in three-dimensional space. So it will be modeled as a mathematical knot quite naturally. So we use the theory of knots to study these reactions. So site-specific combination will be modeled as a two-step reaction, and this is the original idea by Ernst and Sumners, where the substrate is some unknot or link, is incubated with some enzyme, some recombinases and or accessory proteins, well, not and accessory proteins or not, and then that produces usually a distribution of product topologies, sometimes a unique product topology. We want to know what's happening in here, and we want to model each one of these two steps. What are the two steps? Binding and strand exchange. So DeWitt and his former student Klaus set up the basis for this model, and they were inspired by a picture just like this one, and Isabel showed us another version of this picture from the Johnson lab, I think, where you have the enzymatic complex right here. In my picture, we have two emanating arcs, and this is a TN3 resolvase complex. And in Isabel's case, there were, sorry, did I say three? So here there's two emanating arcs, 1 and 2. And in her case, there were 3 emanating arcs. So the idea was to model the enzymatic complex as a 3-dimensional ball, which may be deformed, of course, to look like the protein. And the enzyme and the bound DNA is that ball with 2 strands inside, in the case of this picture, 3 strands inside in the case of Isabel's picture, or 4 or 5 or 6, depending on what the enzymatic complex you're studying. Okay, so here, since I'm doing XER, 2 string tangles. So the enzyme plus the bound DNA is a ball with 2 strands. That is called a 2-string tangle in mathematics. So we use tangles to model this recombination reaction, and the 2 steps will be modeled as a system of 2 so-called tangle equations, where the first step is numerator of O plus P, I'll explain what that is a substrate on knot. And the second step, numerator of O plus R, is a product topology that we know from the experiments. And the unknowns are tangles OP and R. So what are these OP and R? What is this N? So this is, these are the assumptions. So E will be the ball with the 2 strands. This is called the enzyme tangle, and it's that blue tangle up there. And here we're assuming that the substrate is unknotted, so the loops outside are unknotted. If they, untangled. If they were tangled, we could still push them in and add 1 more tangle inside the blue enzymatic tangle. But let's just keep it simple. The outside is untangled, and therefore this construction is the same as the mathematical construction numerator of the tangle E. That's N of E means this picture. And E is the enzyme plus the DNA. So now we're going to, since I'm the one doing the model, I'm going to say, okay, let me look at the core regions of the recombination sites. Where are they? They're inside E, and they're very, very short DNA sequences. I'm going to entrap those inside a little tangle, and I'm going to call that tangle P, the parental tangle. Okay, so the initial conformation, the DNA and the enzymes are fixed. E is written as the sum of 2 tangles, so this is first mathematical assumption, and it is a strong assumption. E can be written as O plus P. Essentially, what that means is take the ball P and partition it. Just take a slice out, and that slice will contain the core regions of the recombination sites. And in my case, this is very, very small piece of the big blue tangle. And then everything else has all that interesting geometry that is trapped before recombination. That lives inside O. So O is the outside tangle and remains fixed during recombination. P contains the core regions of the recombination sites. and P is turned into R. So this is a second assumption of the tangle method. Recombination goes by what is called tangle surgery, where P goes to R. So we can see it here. This is O. O remains fixed during recombination. P is converted into R. So something very simple. So, okay, so now we have everything we need. We have a cartoon picture for the recombination reaction based on experimental data. From this picture turns naturally into a system of two tangle equations, which you now understand. And O, P, and R are two-string tangles, and they're unknown. We want to solve for them. Most tangles in biology are rational, and Isabel gave a lot of examples. And what this means, so in mathematics there are three types of tangles: locally knotted, rational, and prime tangles. So some of the tangles for the mu transpososome case were prime, but those were not really biologically reasonable. So in general, the tangles that have been seen are rational or sums of rational tangles, which are prime, but they're nice prime tangles. They're the Montesinos tangles that Isabel was talking about. And so basically a rational tangle, just for you to have a picture on how to create one of these, you have a tangle with two straight arcs, and you twist the arcs in a smooth way just smooth the formations of these arcs. And you can do horizontal twists, vertical twists as many times as you want. And there's a theorem that classifies these tangles, and it's due to Conway, that says that there's a one-to-one correspondence between rational tangles and the set of extended rational numbers. Extended means that there is the point at infinity, and infinity corresponds to this tangle right here. And then any other tangle can be uniquely associated to one rational number. And this illustrates how we do that. So what is wonderful about this theorem is that it allows us to say, okay, I can tie any tangle by a sequence of vertical twists and horizontal twists. And that sequence I can encode in a vector, canonical vector. I can always ensure that the vector has the same handedness in the twists. So in this case, the 5 means that I started with this tangle and I did 5 vertical twists followed by 2 horizontal twists. But I could keep going on and on and on. Okay, and then from this vector, I can obtain the continued fraction calculation and get the rational number for this tangle. Okay, tangle equations. Isabel also mentioned that if A and B are rational, then numerator of A plus B is a 4-plat or rational knot or link. And these are also very nice, very well studied, classified. We know a lot about them. So now our aim is, if we have an equation, numerator of O plus P, so inspired by biology, equal K, where K is a substrate or product of recombination, P contains those recombination sites, so I can assume that P is very simple, then can we solve for O? And well, the answer is yes, and this is a very simplistic way to put it. There is a lot of theory underlying this, but the quick answer by Ernst and Sumner is that if O and P are rational, then the numerator and the product is a 4-plat written in this way, then P, Y, U, X, V satisfy this very simple equation. So we can solve these systems of equations. So now we go back to the experiment. We have a substrate equation, we have a product equation. We know something about P and R, but sometimes we decide to assume something about them, sometimes we don't. But the real interest is in solving for O, knowing what's all that interesting geometry that is there before recombination. And from the experiments, we know or have a sense of what K0 and K1 are, substrate and product of recombination. So I go to the experiment that Sean presented. XCR recombination of psi yields a unique product topology, the right-handed 4-cat. We know also from experiments in the Sherrod lab by Sean Columns, by Claudia Allen, by Jonathan Bath, that XCR acts only on directly repeated size, that XCR recombination leads to a unique product topology, and that DNA wraps around accessory proteins approximately 3 times. And there's, in this case, 2 copies of Pepe and 1 copy of Argar, like you saw in Sean's structure. So this is translated into a system of tangle equations. Now, If the tangles are assumed to be rational or sums of rational tangles, we can solve the equations, but then we get an infinite number of solutions, so we don't want that. So we want to go back to the biology and ask more questions from the biology to restrict the infinite set of solutions to a finite set that is biologically relevant. So we remark we don't know anything about P and R, so what are we going to do? Well, let's look at these sites. What is P, what is R? So P is 0. Well, P is a tangle that contains the core regions of the recombination site. So the recombination sites consist of a core region of 30 base pairs long and an accessory sequence around 180 or 200 base pairs long. So we leave this one out. We just enclose this core region inside P. 30 base pairs, we saw it earlier this week. DNA is a stiff molecule. It repels itself. 30 base pairs is very short. So you can safely assume that you have the DNA inside this little ball P, and you have two almost straight arcs. They could be bent a little bit, but not very much. You can't have much entanglement in there. So we assume that P is a trivial tangle. See, there are two vertical arcs, two horizontal arcs, and if you look at them from the sides, you could get a plus or a minus. That's a list of trivial tangles. Let me just twist my head so that I see P as two horizontal arcs, like so. So I assume P is 0. And then if P is 0, the sides could be in parallel orientation or in anti-parallel orientation. If P is anti-parallel, then since XCRC and D are tyrosine recombinases, they go through a Holiday junction intermediate. They will well, this is a cartoon of the Holiday junction intermediate reaction right here, and the crossover reaction will take you from P0 antiparallel to R infinity antiparallel. If P is parallel, then the Holiday junction will take you from a 0 to either a +1 or a -1. We can assume that R is what we call an integral tangle, just a horizontal row of twists. Okay, so now we've simplified P and R, and we can reduce the number of solutions from infinitely many to 3, which is really, really great. So just to remind you, this is the data. So this is the first solution we find, and to explain what the solution means, O is -3,0, R is -1. What this means is that the sites wrap around accessory proteins 3 times. The accessory sites wrap around accessory proteins 3 times, exactly 3 times in this model, and the two sites are put together in parallel orientation in the synapse, and recombination turns P0 into R-1, introducing a negative crossing in the domain. The other two solutions predict 5 crossings, the accessory proteins wrapping 5 times around accessory proteins, and here accessory sites wrapping 4 times around accessory proteins, and the corresponding R tangles that all yield the same product topology. Now, there's still 3 solutions. Sean, in his model, he showed this one, so that's the favored solution. So why do we have 3 solutions? So there's a limitation of the tangle method. The tangle method uses a framing of the tangle that forces this tangle to be, well, the drawing to be planar. The tangle is a 3-dimensional object, but you always look at it from a given perspective, and you need to do that to effectively solve the equations. But this is a problem. So we thought, okay, well, let's bring this back to 3 dimensions. Let's not think about the diagrams, and let's think if we can incorporate these 3 solutions in a 3-dimensional interpretation of them. So here we have just cartoon of Pepe, Argar, 2 copies of Pepe, 1 copy of Argar, the psi sites, and then I'm going to show you this movie where we go from the first solution with 3 crossings and parallel sites to the second solution, 1, 2, 3, 4 crossings, anti-parallel sites, to the third solution, 1, 2, 3, 4, 5 crossings and anti-parallel sites. So this is a 3-dimensional object that realizes the 3 solutions found by the tangle method. Okay, so it doesn't mean that this is a correct way, but this is one possible interpretation that could say, well, you have 3 solutions, but in reality they can be interpreted as only one solution. And so we put that together with Sean, with everything he knows about DNA wrapping around R and Pepe in his monster model. and then here there's no crystal structure for XCRCD DNA, but we put here the Cree LOXP synapse that is believed to bind to DNA in the same way as XCRCD. And then, well, we rotated this around the way Sean showed you, and we can indeed get the 3 tangle solutions from this more molecular model. So that was all very nice. Now, at first I said if we assume that the tangles are rational or sums of rational tangles, we can solve the equations. In this case, we don't have to assume it, and this was pointed out by Dorothy in her dissertation, by Isabel in this paper, and I put in the details in the Journal of Molecular Biology paper in an appendix so that biologists can read it. So we made a big effort to try to explain this theorem like step by step, because for mathematicians it's trivial. It's just a consequence of Hirasawa and Shimokawa, and it is. So it's almost not worth proving it. But we did write a proof, very detailed, for biologists to read. So I'm hoping that you guys will read it. So, and basically what that theorem says in biological terms is there's a system of 2 tangle equations taking the unknot to that 4-cat. Then we can prove that without assuming anything on O, P, and R, that P and R, R, are locally unknotted and O is rational. So since O is rational, the solutions we found are all possible solutions under the tangle method assumptions. So we have a complete set of solutions. This is the program that Isabel was referring to. It's called TangleSolve. It implements the tangle method, and it allows us to do processive recombination. For XCR, we're dealing with non-processive recombination. It does compute Montesinos solutions, so I just wanted to point that out. So both rational and sums of rational tangle solutions. And well, that's a reference. Okay, how much time do I have? Okay, good. So now let's go to more sophisticated DNA unlinking by XCRCD FTSK. So that's what I started with in my talk as a motivation. So XCRCD will, so replication creates catenanes or links. They're right-handed torus links with parallel sites, not the anti-parallel like in XCR at psi. Replication makes right-handed torus links, just like those, but with parallel sites. And here the sites are the diff sites. And in this gel, oh, I didn't put a disclosure. There's one gel in this talk. In this gel, from the Sherrod lab, so this is work of Ian Grange and Miguel Brego. They showed how, well, they show here the replication links as a substrate, and then a time course of these links incubated with XCR, CD, FTSK, acting at the diff sites, the XCR-CD acting at the diff sites, and we see how XCR-CD unlink the replication links. This unlinking is nowhere as efficient as Topo4 unlinking, but they can unlink the replication links. And this is the same reaction, but no time course, so 30 minutes at 37 degrees, and the complexity, the topological complexity is pretty much gone. The other thing to notice here is that dimeric knots appear as reaction intermediates. So we wanted to try to understand, okay, so how is this enzymatic complex unlinking the replication links? Can we say something about the mechanism? Can we say something about the pathway? So, like Isabel pointed out, I mean, the biologists know really how to think about this, even without mathematical tools, so they don't need that. They just have wonderful brains, and they say, okay, well, the unlinking pathway has to be this one. It has to be a stepwise unlinking pathway where you start, for example, with a 6 crossing torus link, and you go from a 6 to a 5, 5 to 4, 4 to 3, 3 to 2, 2 to 1, 1 to unlink, all done. Stepwise unlinking, and that's the proposed mechanism. Okay, well, let's see what the math says about that. So experimental data, so let's see how we translate this into mathematics. We have a distribution of replication links, 2n cats, for different n's, incubated with XCR-CDF-TSK. The products are knots or links in a distribution again, but mainly on links. We will model the recombination using our tangles. And let's just try to write the equations. The substrate is a numerator of O plus P, will be a parallel to MCAT. So let's just go one step at a time. Take one of the substrates, parallel to MCAT, in this case the 6 crossing torus link with the 2 sides in parallel orientation, and the product is some knot or link, okay? But we know from the experiment that over time, most final products are unlinked circles. But you see, I mean, this is much more complicated than before. Before, we knew exactly what the product was after one step of recombination. Here, in theory, we have no idea. And we know that after a certain amount of time, you get unlinked products. But is it after 1 step, 2 steps, n steps? We don't know. So everything that I will say from now on is joint work with Koya Shimokawa, Kai Ishihara, and then Ian Grange and Dave Sherratt. So the first theorem that we proved was if we assume each action of XCR-CDF-TSK is modeled by P0, so same assumptions as before, and then R is infinity minus 1 or 1, so in mathematical jargon, by advanced surgery, then at least 2M recombination events are needed to convert the right-handed 2M-cat with parallel leaf sides to the unlink. So we know the minimum length of the unlinking pathway. The shortest unlinking pathway has 2M steps. So for a 6-cat, the shortest unlinking pathway has 6 steps. We can't go from the 6-cat to the unlink in 1 step with parallel sides. With anti-parallel sides, everything changes, okay? So this is one example of that minimal pathway, 2M cat to 2M minus 1 torus knot, and so on and so forth, trefoil, 2-cat, trivial knot, trivial link. Now, the shortest unlinking pathway not only has 2M steps, but it's also unique. So this is also another really nice result. So if we assume that an unlink is obtained from the 2M cat after 2M recombination events, and strong assumption, we assume that each recombination other than the last one reduces the number of crossings. I tend to call the unknot, I tend to say that it has 1 crossing so that I go from 1 to 0, but just to be safe. So we assume each recombination reduces the number of crossings, then every intermediate is a right-handed 2K cat or right-handed 2K plus 1 torus knot, or the unknot. So those are all the possible products of recombination. And then the only possible pathway is this one, where you go from a 2M cat to a 2M minus 1 torus knot. So from a 6 cat to a 5 knot to a 4 cat to a 3 knot to a 2 cat, etc. So so just to, this is the same as before, but just to give a concrete example in case the 2M's are driving you crazy. So if we start with a 6 cat, then the product has to be a 5 knot. If we start with a 5 knot, then the product has to be a 4 cat. If we start with a 4 cat, the product has to be a trefoil. And we just walk our way down until we get to the unlink. So that yields the unique pathway. So the XCR-CDF-TSK complex unlinks DNA by a stepwise mechanism, taking the 6 cat into a 5 knot, the 5 knot into a 4 cat, etc., until the substrate is unlinked. And this is exactly the pathway that you get. Now, if we assume that recombination is iterative, because these enzymes seem to be binding the DNA, doing a recombination step, resetting, I mean, it's not processive in the way of serine recombinases, but because of the holiday junction, but they seem to be resetting and doing another recombination step and not letting go until they've managed to unlink. So if we assume that the recombination is processive, then we can show that O is rational and that this is a topological mechanism of action. But this iterative assumption makes some people uneasy, so we don't need it. But if we have it, then we have a stronger mathematical result. Now, if we assume for the 4-cut, if we assume rationality of the tangles, then we get 3 mechanisms of unlinking the 6-cut into a 5-knot. So now we look at the tangle equations. N of O plus P is this 6-cut, N of O plus R is this 5-crossing knot. We assume that P is 0, and then we obtain 3 solutions like we did for the other case, where R is plus 1 here, R is infinity here, and R is minus 1 here. So very similar to what we had before. So I have another movie for you. We can, assuming it's going to work. I don't know why it didn't start. Uh-oh. Okay, well, maybe at the end of the talk I can show you the movie. So I checked the other one, but I didn't check this one. Well, but you can imagine what's going to happen. We're just moving this rigidly in 3-dimensional space, rotating it around without cheating, without stretching or pulling anything. And this is one solution from the tangle equations, 1, 2, 3, 4, 5 crossings here, and then 1 crossing here. Here we see another crossing and the 2 sides in, what is this, in parallel orientation. And then as it moves around, it circulates. So that's this solution here. It circulates around the other 2 solutions. So again, we can find a 3-dimensional object that realizes all these 3 solutions. We can do this at each step of the pathway. So we have this pathway, and I just showed you this step right here. Well, we can solve tangle equations at each step. For the last 3 steps, we can prove that the tangles involved are rational and find unique solutions. After the 3rd step, we can't prove that the tangles are rational, so we can solve the tangle equations using the tangle method, but there might be other funkier solutions that we don't know about. But in every single case, I see 3 solutions, and in every single case, I can put those 3 solutions as rotations of the same object. Okay, I don't have much time to go through that, but if anyone is interested, we have a figure that incorporates the 3 possible solutions in the pathways. So to conclude, XCRCD at psi converts unknots into 4-cuts in a unique manner, and we have this molecular model. XCRCD FTSK at diff on link replication links in a stepwise manner, taking 2n torus link to 2n minus 1 torus knot, etc., until getting to the unlink. The pathway is, the shortest pathway is unique, and is consistent with a unique topological mechanism of action, with certain model assumptions that seem to be supported by the experimental data. Now, what are we doing now? We're analyzing other possible pathways. So we have that assumption that at every step, recombination takes the complexity down. The crossing number goes down. Well, let's not assume that. Let's think maybe that's too strong of an assumption. And we don't have the experiments to show that it's indeed going down. We see the experiment, the trend is down. So in the gel, the trend is upward. Everything is being unlinked. But at each step, is it being unlinked at each step? We don't really know. So let's not assume that. So if we assume that the complexity remains the same or goes down, then there are other possible options. So these are other pathways, and this is also in collaboration with Koya and with Kai and Masaki, Koya's student. And these are other possible pathways of recombination that can take you from a 2n cut to the unlink. And we're also interested in studying the frequency of each one of these pathways. So we remove that assumption, we get more possibilities. Which possibility is the most likely? So can we do that using Monte Carlo simulations? So we're doing that. So here you see an 8-crossing torus link converted into a 7-crossing knot, 6-crossing link, 5-crossing knot, eventually 4-crossing link, trefoil, Hopf link. It's taking longer. And then the unknot at some point, and then eventually the unlink. So we're calling this recombo. Recombo identifies 2 recombination sites, performs recombination, and can help us to give probabilities to these, each one of these transitions in the pathways. Okay, so there are your 2 unlinked circles. And then finally, in the last minute I have, 3D considerations of the tangle method. I've been talking about this for years. Isabel has been talking about this as well, and she talked a lot about it today, so I was very happy to hear that. Well, I mean, there's this problem, right? We get 3 solutions, but we could interpret it as one. So let's define equivalence classes. Let's do something in 3D. A few years ago, talking to the WIT, he said, well, maybe we can put things in a tetrahedron and study the symmetries of the tetrahedron. So we started doing that. So this is from the WIT's old paper. But so we put things in the tetrahedron and just look at the symmetries of the tetrahedron and see for simple tangles, this symmetry will take a tangle with 3 crossings to a projection with 2 crossings. So the tetrahedron is too rigid, so I can't explain 3 different projections. I can only explain 2 with a tetrahedron. So maybe we need something more sophisticated than a tetrahedron. Maybe we just need to do it in the sphere, but in a smart way. But, well, we're working on this. And this is work of my student who just graduated and went for a PhD at UC Davis, or she's starting right now, Katrina Wono. Okay, so acknowledgements. All this started with the WIT during my PhD a long time ago. And also during my PhD, I met Dave Sherratt, I met Sean Collins, and we started working on XCR at Sci. Later, I spent a summer in Oxford and interacted a lot with Ian and worked on the unlinking by XCR. Then I have a wonderful mathematical collaboration with Koya and Kai, which has really enriched what we can do. All the wonderful pictures and movies are thanks to Rob Sharine and Notplot, students who have worked at one time or another in the software, the TangleSolve, and also different aspects of the project. And I should have Katrina there, and just my funding. Thank you very much.
So we have time for 1 or 2 questions. And yes, Steve.
So, Mariel, it's very inspirational work. Just a comment, really, I think. If you have multiple competing pathways, the pathway that's going to win is going to be the one that wins kinetically. And unfortunately, that sort of information isn't going to come from the kind of equilibrium statistics that you're doing, that we're doing, and other groups perhaps. What you really need are rate constants. And so experimental biologists will need to do a better job on the kinetics, because we can get ratios of rate constants between 2 topological states, but we're not yet at a point where we can compute from first principles what those rate constants are. So this is kind of a general appeal to the community to start thinking about those issues, because I think the question of multiple competing pathways is an important one, and we're not there yet.
That's a great comment. Thank you. Any other questions?
So before thanking again Mariel, I wanted a little bit to mention this award which Mariel received recently. So that's a presidential award for most talented scientists of young generation. And so Mariel, we are thankful we are congratulate you, and we also thankful for you because you little bit, well, not little bit, you really contributed to that this field we are working in is more known in the field. Thank you very much, Mariel.